Alright boys, so today, first of all we are going to learn about oligosaccharides. In the last lecture we have talked about the monosaccharides and we learned that mono means one, saccharide means unit, right? These are the simplest carbohydrates, right? Because single molecule is present in one carbohydrate. Now oligosaccharides, they can have 2 units up till 10 units joined together. Means that these oligosaccharides, they can be disaccharides, means 2 monosaccharides joined together, trisaccharides, butesaccharide, pentasaccharide, right? Hexasaccharide, heptasaccharide, like that, right? In your syllabus, we have only the disaccharides. Now, disaccharides again, these are 2 words. Di means 2 and saccharide means sweet units. So what is there that the 2 monosaccharides, 2 sweet units, when they join together, they make a disaccharide. We have many different examples of the disaccharides. One of the most common example is maltose, right? Another common example is sucrose. Another example is lactose, right? Now first of all, we are going to talk about maltose. Now maltose is a disaccharide in which 2 molecules of alpha glucose are joined together. 2 molecules of alpha glucose are joined together. Now today we are going to also learn how these 2 molecules of alpha glucose join together. Here I have a question. When 2 monomers join together and they make a dimer, what is that kind of reaction called?
Condensation.
Very good. Condensation reaction, right? So today we are also going to learn the condensation reaction by this one maltose molecule is being made. And when this one maltose molecule is made, what kind of bond is in between 2 glucose molecules, we are also going to discuss that. For that purpose, I am going to draw the structure of glucose. Now, if you guys remember, and you guys have practiced for the structure of the glucose, this is the alpha glucose. Do you remember? Right? So this is one alpha glucose molecule. Now this one alpha glucose molecule has to join or react with the second alpha glucose molecule. Let me draw another one. Remember that on the 3rd carbon, the hydroxyl is on the inner side. Right boys? So these both are alpha glucose molecules. Now what will happen? That there will be reaction between this hydroxyl and this hydroxyl, right? Now the hydroxyl from here and hydrogen from here, they will both join together, and this is hydroxyl, this is hydrogen. What will get? One molecule of water will be removed. One molecule of water will be removed. And I can represent that removal of one molecule of water by minus H2O writing on the arrow. I can represent it in another way. I will tell it later, inshallah, right? Now, how I'll show this compound now, very easy, very easy. First of all, what you do, you again draw both of these glucose structures, but you don't draw these things, right? Now, look here. Then I will draw this one. Now we have all of these things here, like this. Here we have now look here. This hydroxyl and this hydrogen join, right? What is removed? What is left behind? This oxygen. Now what will happen? This oxygen will come in the middle and this will be joined over. Understood now? And remember, these hydrogens are still there. Right? Now, this bond, which is being made between two glucose molecules, it has a name. It has a name. How we are going to name that? First, come back over here. Let's name these numbers of carbons. We know that. We have already numbered this before. 1, 2, 3, 4, 5 and 6. Do you remember this or not? 1, 2, 3, 4, 5 and this 6. Now, the first molecule taking part in this chemical reaction is what? An alpha molecule, right? So I will write over here alpha. And over here, one of the carbon taking part in the chemical reaction is carbon number 1. Second carbon taking part from the second molecule is carbon number 4. So I will write 1, comma 4. Right? Carbon number 1 and carbon number 4. Carbon number of 1 glucose and carbon number 4 of the second glucose took part in the chemical reaction, right? So alpha 1, 4, and this bond is called as a glycosidic bond, or we can also write it as glycosidic linkage. So we will name this bond as alpha 1, 4 glycosidic bond or alpha 1, 4 glycosidic linkage. Alright? So this is what? These are two monosaccharides, glucose molecules joined together. So this is actually structural formula of what? A maltose. Now maltose is very important disaccharide. Maltose is very important disaccharide. I am telling you right now, I will ask this again from you guys. You guys have learned this thing before. I have a question. What is the name of the polysaccharide that is stored in plants? Who remembers? Yes.
Starch.
Excellent. Starch. Next question. There is an enzyme which is present in our mouth, produced by the salivary glands, that digest starch. Excellent. Amylase. Next question. When amylase digests the starch, what is the product that we get? Maltose. Understand? So you have to remember, starch is being digested by the amylase, and the product that we get is this. You only heard the name before. Now you know what is it. Right? Another thing, that maltose is also very important in some beverages as well. Now, this is Saudi Arabia. It's a Muslim country, right? We do have beer over here, but that's non-alcoholic beer, right? When you buy that beer, non-alcoholic, what's written on its sticker? Malt beverage. because it is prepared from maltose. What do? They take starch from different sources. They can take the starch from the corn, or they can take the starch from the wheat, or they can take the starch from barley. Right? They have usually made barley over there, right? They treat it with amylose. When it will be broken down into maltose, then they will ferment it. Understand that?
Yes.
Then they will produce that malt beverage. Alright? So that is this maltose. Next, let's talk about sucrose. Again, sucrose is also a disaccharide. Now remember one thing, that these disaccharides, they have become complex than monosaccharides, right? So they are not sweet. They are not sweet. But sucrose is an exception over here. There is a reason behind it. There is a reason behind it. Before I go into that detail of that reason, lactose is also a disaccharide which is present in milk. It is also called as milk sugar, right? Now sucrose, it is made from 1 unit of alpha glucose and 1 unit of beta fructose. 1 molecule of alpha glucose when joins with the 1 molecule of beta fructose. Alpha glucose and beta fructose, when they join together, they make 1 molecule of sucrose. Understand? While lactose is made when 1 molecule of glucose joins with 1 molecule of galactose. Now, galactose structure is also a bit different than the glucose, right? The chemical formula, empirical formula is exactly the same, right? C6H12O6. Only the difference is there that, you see, in the beta glucose, what did we discuss? Hydroxyl is up, hydrogen is down, right? In galactose, with this change, there is another change. This hydroxyl is also up and this hydrogen is also down. So there is shifting of hydroxyl going up is at 2 position. Position number 1 and also position number 4. 4. Alright? Now, I am going to show you guys the glycosidic reaction, this reaction between alpha glucose and beta fructose. Alright? Now this is also very important, so please pay attention. Again, first I am writing down the glucose. And remember that this is what? Alpha glucose. Right? Now, it reacts with fructose. Now remember that fructose is a 5-cornered ring. Did you guys do the practice of this?
Yes.
Alright? Now, we know that this is what? This is alpha glucose. And this one is beta fructose. Right? Now what will happen? That this hydroxyl will react with this hydroxyl. This OH will join with the hydrogen. There will be removal of 1 water molecule. There will be removal of 1 water molecule. Now, next step, again, we have to join both of them and write them together. Simply, leave this middle part in the beginning. Write these both formulae over here and then join them at the end. Don't make it too much complicated for you guys, right? So, what do we do? Now what do? I join them together with, let me use some other color, red one. Join together. Alright? Now let's complete the valencies. Here we have hydrogen. Here hydrogen, hydroxyl, hydroxyl, hydrogen, hydrogen, hydroxyl, hydrogen, 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 hydroxyl, hydroxyl, hydrogen. Understood now? Now what has happened? That a glucose molecule has joined with what? A fructose molecule. Now, the first molecule in this chemical reaction is what? Alpha glucose, right? So we will take the alpha from here. Right? Now again, do the numbering. This is, let's use some other color. 1, 2, 3, 4, 5, 6. This one is numbering starting from here. 1, 2, 3, 4, 5 and 6. Why the numbering is starting from here? This hydroxyl group is over here, which can take part in chemical reaction. Right? Why this hydroxyl group is over here? Because this was actually the functional group. Remember? Let me draw over here very quickly the structure of fructose. 1, 2, 3, 4, 5, 6. This was the functional group, right? I told you that it comes and binds over here. Remember that or not? So this O changed into what? OH. This O changed into OH. That is why. And remember I told you, we start the numbering from the carbon which is nearest to the functional group. So we started the numbering from here. 1, 2, 3, 4, 5 and 6. So this oxygen changed into hydroxyl. That is why we are starting the numbering from here. See, this is the straight chain structure. I told you when it is in the solid form, undissolved form, it is straight chain. When we dissolve it in water, it changes into ring structure. Right? For changing into the ring structure, the functional group carbon attaches with the second last carbon. When it will attach with the second last carbon, this is C double bond O, right? It will become C single bond O, and with this one H will come. And it will become what? Hydroxyl, this part. Understand? And with this is CH2OH, 1. And this is also CH2OH. So if you see, there are two tails. One this one this one. Alright? So this becomes ring structure. So this oxygen became the hydroxyl group. So that's why we started the numbering from here. Because it was nearest to the functional group. Now, in this case, what is happening? First carbon, let's see how already know. First carbon of the alpha glucose took part in chemical reaction. We write down 1, comma. And second carbon of the beta fructose took part in the chemical reaction, right? So we write comma 2. And when these two monosaccharides join together, the bond is called glycosidic bond. So we will name it as alpha 1, 2 glycosidic bond or linkage. Understood? So this is the fructose, this is the sucrose. Now, if you guys remember, when we were learning about the monosaccharides, we learned that the monosaccharides are sweet in taste, right? But there is one thing, that when there is a keto group, when there is a keto group, its physical property is that it is more sweeter than an aldehyde group. It is more sweeter than an aldehyde group. That is why fructose is sweeter than glucose. Right? Those fruits that have fructose in them, they are more sweet. They are more sweet. So, when there is glucose attached with fructose, it will give what? Sweeter taste. But not as sweet as glucose. or fructose, but still sweet. But maltose is not sweet at all. When you drink natural milk, is it sweet? No. Right? Either it has glucose and galactose in it. It becomes complex. It becomes disaccharide. Understood, everyone?
Yes.
Now, at home, you will draw these 3 times each, and you will make the structure of lactose. You will make the structure of lactose. Now, I have already drawn the structure of glucose over here. For you, I am going to draw the structure of galactose here. Alright? So that you can use this and you can draw the same kind of chemical reactions. So this is what? This is galactose. Alright? Now remember, in mathematics, you practice to become perfect, right? Same, in these kind of chemical equations, in these kind of reactions, you practice to become perfect. Understand? So you practice a lot. I want to see when you come back on Sunday, all these reactions. 3 times each. And also the reactions for the lactose. Alright? So this was about the disaccharides. We can take this picture from the screenshot, right? Later. Now I'm going to rub this and we are going to talk about polysaccharides.
How is lactose made? By adding
Glucose and galactose. And there will be 1,4-glycosidic linkage. I am giving you hints already. As it is an assignment, you can take help from the internet as well from home. Alright? So now, we are going to talk about polysaccharides. Again, 2 words. Poly and saccharides. Very good. Poly means what? Many. And saccharide means sweet units.
Sir,
Yes.
can I take Hamza Salman for 2 minutes?
No. After this class.
No problem.
Thank you. So, polysaccharides. Poly means many. Saccharide means sweet units. Now, remember I told you, monosaccharides have 1 sweet unit. Oligosaccharides have 2 to 10. If there is any molecule has more than 10 monosaccharides joined together, that will be called a polysaccharide. Simple. Right? More than 10 monosaccharides joined together will be considered as polysaccharides. Now polysaccharides have become more complex than oligosaccharides, right? They already in the form of oligosaccharides, they lost their ability of sweet taste, right? So polysaccharides are not sweet at all. Another thing which we talked about before, that the monosaccharides, they are soluble in water. Do you remember that or not? I told you, glucose is a polar molecule. Why glucose is a polar molecule? Next topic will explain it, inshallah. When we'll talk about the structure of water and we'll talk about dipoles. Remember me, remind me that we have to talk about dipoles, right? You will learn about that in chemistry as well, inshallah. But I will tell you right now. Maybe chemistry will come later. Even the oligosaccharides, the disaccharides, they are also soluble in water. because they are not too much complex. But polysaccharides, they are totally insoluble in water. Now, in your home, you have flour, right? Your mother sometimes, she makes the dough. Does flour dissolve in water?
No,
It's starch. It does solidify. It does not dissolve in water. Right? So it's simplest example. You have rice in your home. You boil them. Do they dissolve in water? No. Right? That is starch. Starch is a polysaccharide, right? Now, there are many examples of polysaccharides. Starch is the most common example we talk about again and again. Another example is glycogen. Right? Another example is cellulose. Now, anyone who will tell me the scientific name of starch? Who remembers? You have told me the enzyme that breaks down the starch. And remember that the enzymes are named after their substrate names. This is a type of starch. Amylose. Remember, when we started learning about carbohydrates, we learned one thing, that their names ends at -ose. So, the scientific name of this starch is amylose. There is amylopectin as well. We are going to talk about both amylose and amylopectin. Now, you guys know that this amylose and amylopectin, this is present in plants. It is the storage food of the plants. It is the storage food of the plants. Plants store their food in the form of starch, which is amylose and amylopectin. Now, you guys know when the photosynthesis takes place, what is formed? Glucose. Then glucose is transported from the leaves to the roots. In which form the glucose is transported?
Sucrose.
Excellent. Sucrose. Question is, why is it transported as sucrose? Why not just glucose?
Less reactive.
Very good. I taught you that. And complex than glucose. Glucose is monosaccharide, right? It can easily take part in chemical reactions. But when it will become disaccharide, it will become complex, cannot take part in chemical reactions, will not be used up during its pathway, right? During its passageway. And most importantly, that sucrose is also soluble in water, just like glucose, so easy transport. Next, this glycogen, it is present in animals and it is the storage food for the animals. You have learned in our body, when we eat starch or glucose or anything sweet, that is digested in our body, absorbed into the small intestine, into the blood, and taken to the liver. I have a question. This will come, we have circulatory system chapter in this book, in this syllabus of AS. What is the name of the blood vessel that carries the food, digested food, from the small intestine to the liver? Who remembers? Excellent. Hepatic portal vein, right? It will take to the liver. In the liver, what happens? The glucose is changed into, right? And this glycogen is stored at two places in our body, in muscles and in liver. Very good. Understand? Then, this cellulose, it is the material that makes the cell walls in the plant cells. Now, let me explain that this amylose, amylopectin starch, it is made up of alpha glucose. All the monomers, they are alpha glucose. In glycogen again, all the monomers, they are alpha glucose. But in cellulose, all the monomers, they are beta glucose. And I have taught you the structure of the beta glucose. Now, first of all, I am going to tell you the structure of amylose. Then we'll discuss the structure of amylopectin and glycogen, because the structure of amylopectin and glycogen is a bit similar, right? And then we'll discuss the structure of the cellulose, inshallah, right? So let's first of all talk about the structure of amylose, simple straight chain. Now, we can simply draw the structure. This is we can also represent it like this. First of all, draw these hexagons, right? About 3 or 4 of them. What? How do I remember, or how will you remember? By practice. Alright, guys?
Yes, sir.
Now, next, what do? Simply join them by oxygen. We can make like this. Right? And we can show that this chain is going forward like this, and this chain is going forward like this. Right? Like sometimes what do? We put these dots like this, right? To show that this chain is going in this way as well and this way as well. We can complete rest of the right? So this is what? All of them have what? Alpha 1,4 glycosidic linkage. What? This is not V. Let me make it simple for you. Don't make it complicated. It's simple. This. We learned about this before? Okay. Make it simple. Now it's okay? So this is how these are joined together. We can show like this. So all the bonds,
4 types.
it's up to you. You can make 5. You can make 3 only. Right? Main thing is to show that what has happened, that all these, they have joined together to make a long chain. Right? This is alpha 1,4 glycosidic linkage. This is alpha 1,4 glycosidic linkage. Now, actually when I drew the bond like this, to show that there is a bond angle. There is a bond angle. And that bond angle does not keep this chain straight. Does not keep the chain straight. Because of that bond angle, this chain will what? Bend. Chain will bend. So starch does not have straight fibers. Starch does not have straight fibers. When these alpha-1,4 glycosidic linkages are made, at every linkage there is an angle. There's an angle that makes it bend. So that's why starch is not a structural protein. It is not a structural protein. It is not used in making any structure. Right? Cellulose is a structural protein. Remember that?
Yes.
Because it makes a structure, cell wall. Because its chain is straight. We'll talk about it inshallah, right? Another thing guys, when we test the starch, here I have a question. You are very new, right? You just finished your O levels, right? So you know these things. You should not forget these things because these are going to help you this year, next year and rest of the life as well. Inshallah. What is the name of the test for detection of starch? Iodine test. Right? Now, what is the result of the iodine test? Blue black. Right? Blue black, right? So when we take the starch and we add 1 or 2 drops of iodine solution on it, now I have a question. What is the color of iodine solution?
Yellowish brown.
Very good. Yellowish brown. So yellowish brown color changes into blue black color. Understand? So remember this. We will inshallah perform this test over here, right? In the coming week or next inshallah. Next wise I am going to draw the structure of amylopectin. Now amylopectin and glycogen, I told you they are closely related structures. Because these are not simple straight chains. These are branched chains. These are branched chains. Now, let me tell you that amylose, it has only alpha-1,4 glycosidic linkage. Amylose has only alpha-1,4 glycosidic linkage. But this amylopectin and glycogen, they have alpha-1,4 glycosidic linkage and alpha-1,6 glycosidic linkage.
These are for amylopectin.
Amylopectin and glycogen both. Amylopectin and glycogen both. They have obviously alpha-1,4, but they also have alpha-1,6 glycosidic linkage as well. Alright? So now, I am going to draw the structure of amylopectin and glycogen. Now there is an easy way to draw the structure as well. There is an easy way to draw the structure as well, where you guys don't have to write these carbons and these hydrogen and hydroxyl. Right? But I will not recommend that. I will recommend this because this is how you will become perfect. You will know where which atom is there, where which element is there, right? Now I am just going to draw a small chain. This is the representation of one glucose molecule. This is the representation of one glucose molecule. Each corner where nothing is written is represented by a carbon. Each corner where nothing is written, it is mentioned, it is considered as carbon. Right? So what do? I draw another one over here. Now, all of them, they will be joined by how? Alpha-1,4 glycosidic linkage. Right? Adil, this is where you have to pay attention now. Now, what I am going to do, I am going to draw another chain over here, down here. Right? So let me draw that chain in the red color. Now, let's see that these chains are joined with each other, right? Now, remember over here, I am going to show the bonds how they are made over here. I am going to show you guys alpha 1,6 glycosidic linkage now. Remember this, that this is the carbon number 1, 2, 3, 4, 5 and this is 6. Understand? And on carbon number 1 there is hydrogen over here, hydroxyl over here. Understand this or not? Say this is carbon number, let me use the other color, let me use the blue. 1, 2, 3, 4, 5 and this is the 6th carbon CH2 OH. And remember that over here we have hydrogen, hydroxide. Now also remember this is what? Alpha 1,4 glycosidic linkage. Remember this as well. What is this? Alpha 1,4 glycosidic linkage. Now these are two chains, two complete chains with alpha 1,4 glycosidic linkage. Now what will happen? Look here. This hydroxyl will join with this hydrogen. You see that this hydroxyl will join with this hydrogen. And what will be done? Minus H2O. One water molecule will be removed. Now when the water molecule is removed, instead of writing the whole structure again, what I am going to do, I am going to show it in the same diagram. What will happen? That this hydrogen and these hydroxyl are gone as water, right? This is gone as water. And this oxygen will come where? In the middle over here. Here. Got it? The oxygen which was with this CH2OH will come here in the middle of these two. Now look, this is the 6th carbon. This is the 1st carbon. Now the bond is being made between 1st carbon and 6th carbon, right? Now this bond will be called as alpha 1,6 glycosidic linkage. What it will be called? Alpha 1,6 glycosidic linkage. Let me explain this again. I will rub it and I will draw it again. I will show this all again. Now remember that over here is hydroxyl. Remember that or not? This is a glucose molecule, right? So it has hydroxyl over here. Then over here, this is CH2OH. This is also hydroxyl over here. Zamin, leave that, draw later. Pay attention over here first. Glucose molecule. Have hydroxyl over here, right? Let me draw a glucose molecule again on the side for your reference. H, OH, H, OH, OH, H, H, OH, and H. So look here, this is the hydroxyl over here. Can you see? And this hydroxyl is over here. Can you see? Right? This is separate chain, this is separate chain. What will happen that,
So what are oxygen?
Sorry? sorry,
Oxygen.
what oxygen?
So that you have drawn there. Oxygen. It starts under oxygen. Amylose.
Yes, because I am showing that this chain is going forward. There is another glucose molecule over
Why
here.
this is not going forward?
Because here it is going to make a bond over here.
Sir,
Right? This chain is extending in this direction. Let me show like this. This chain is extending in this direction. Alright? This chain can also extend in this direction. We'll talk about it. But please pay attention till here. Don't become, you see, too confused about it. This is the glucose molecule right in front of you. This is carbon number 1, 2, 3, 4, 5 and 6. At carbon number 1, do you see a hydroxyl group? This is carbon number 1, you see a hydroxyl group. This is carbon number 6, do you see a hydroxyl group? This is the carbon number 6, do you see a hydroxyl group? Alright? Now, there will be condensation reaction between the carbon number 1 of this chain and carbon number 6 of this chain. Alright? So what will happen? Let me show you that this hydroxyl and this hydrogen, they will be lost as what? One water molecule. Gone? What is left behind? This oxygen. What is left behind? This oxygen. Now this oxygen I am going to show here will come in the middle as what? As glycosidic bond. Alright? As we learned in disaccharides, right? Before the bond was between 1 and 4, right? Here bond is between 1 and 6. So this will be called as alpha 1,6 glycosidic linkage. Yes, Adil, you have a question.
can I ask, is it only connected to one point?
Yes. Now, look here. This chain, does it have hydroxyl over here?
Yes.
It can extend in this direction as well. It can extend in this direction. Now what has happened? This is not one chain now. These are two chains connected. These are two chains connected. There can be another connection somewhere over here. There can be another connection somewhere over here. It has become more complex. It has become more complex. It's not one chain now. It's more than one chain connected, joined together. So the amylopectin and glycogen, they have the structure like this. Glycogen and amylopectin, they have structure like this. Understand everyone? So this is the amylopectin. Understood everyone? Okay. Next, we are going to talk about cellulose. Now, I have taught you the structure of beta glucose, if you guys remember. Right?
Yes.
Now, I will draw the cellulose structure over here and then I will explain in detail, right? But first of all, let me write that this is what? This is alpha glucose. Now, let me write for you guys the structure of beta glucose so that you guys don't become confused. So this is what? This is beta glucose. Now the drawing the cellulose structure is a bit tricky, but I have confidence in you. You can do it. Right? If at this age I can do it, then you guys can do it. Now, look here. First, I'm going to tell you, just draw the units. Just draw the units. Don't make the bonds first. Right? What do you do? This is straight. Now, if you want to make it easy, do this. Carbon, carbon. Right, one carbon here, one carbon over here. Then do this. This is upside down. This is the same glucose molecule but upside down. Do you see? Oxygen is over here. Carbon, 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 carbon. And with this is the sixth carbon. Do you see? Right? Next, you do this. Later I will change the color of these bonds. Right? Now again what you do? Right? One carbon here, one carbon over here. Then what you do? Look here. Carbon, oxygen like this. Right? Now it has come back to its original position, right? Next one is again upside down. Carbon, carbon. Next you do carbon over here, oxygen over here, like this. Carbon, carbon, CH2OH. Now, I am making this, can go like this, right? And here I am making this, can go like this. Now let me change the color of the bonds for your understanding. Period is over? Just give me a few minutes, okay? Because this is very, very important. This is the last thing which we need to discuss, then the period will be finished. Now look here. Let's do the numbering. Let's do the numbering. You have to write down this hydrogen hydroxyl, you will do later, right? For me. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Now if you see, these are beta glucose, right? So what we will write? Alpha or beta? We will write beta. The bond is between first carbon and fourth carbon, right? 1, 4. Alright? And what is this? Glycosidic linkage. Now, listen. Because of twisting of each beta glucose molecule, alternatively, the bond angle do not change. And a straight chain is formed. A straight chain is formed. As the straight chain is formed, it is in the form of fibers. It is in the form of straight fibers. That is why it can be used as what? As a structural polysaccharide. That is why it is used as what? As cell walls building material. Understood, everyone? We'll continue from here. Inshallah, next class. Take care and Allah Hafiz.